Department of Chemistry at the Stanford Neuroscience Institute at Stanford University. She has an extraordinary list of accomplishments and grants and awards from her impressive career. And her talk today is the role of membrane curvature at the nano-bio interface. So Dr. Cui, if you wish to take over.
Thank you. And it's my pleasure to, in this special time to give a talk about my research to all of you. And thank you for participating in this one. So today I'm going to talk about is, what our understanding of how membrane curvature plays a role at how cells interact with nanomaterials. So in actually many situations, I would say in health and in disease, we encounter situations where cells directly contact with synthetic materials. That including you have bone implant, like many of the times a titanium bone implant, knee replacement, a dental implant, and more recent study including deep brain stimulation. Then you need to have electrode implant or brain machine interface. Again, you need to have electrode directly contact with neurons. Or as simple as if you're a graduate student or even undergraduate student, you probably have done cell culture. And in those cases, what you're doing is you're growing mammalian cells, in those cases, on top of plastic. So in all of those situations, what we encounter is an interface directly between biological cells and synthetic materials. In this image particularly, this is actually an SEM image of a titanium implant. This is a picture directly from a company. So that means it's currently this is a titanium implant that's going into a patient. So in this specific case, if you look at the image here, it's not a typical metal titanium surface. If you look at surface, it's very rough. There's a lot of peaks and valleys and quite random features. So one question is, why do companies do this? Why the companies do specific surface treatment to make the dental implant or titanium implant very rough? The reason is, if you look at all different companies that make those implants, they have a variety of techniques. Some use, sanding, as simple as sanding. Some using, bombard the surface with high energy particles to create those rough surface. The reason is there are clinical studies to show that if an implant have a rough surface as compared with a smooth titanium surface, usually the body accepts the implant better and is less likely to induce immune response and failure of the implant. So it has been actually many years, people trying to understand why cells in this particular cases, cells integrate with rough surface better than smooth surfaces. Rough surface, irregular shape are usually difficult to study and the mechanisms, but nanofabrication has thus allowed us to fabricate precisely controlled topography, such as nanopillars, fibers, those nanogratings, pores, nanopores. And those different topography, instead of the randomly shaped or randomly arranged rough surface, allow us to understand what specific topography cells prefer and what is the fundamental mechanisms. I would like to say that this is a rather large field, particularly for mechanobiology and biomedical engineering field. Those are the examples I gave. There's certainly not exhaustive list. There are a lot of lists. Those are the groups mostly working on the nano sensors, nanostructure sensors, particular vertical nanostructure sensors I'm going to talk about today. And to talk about the nanostructure surface, I want to start with an unpublished result. Here you will see that on the left surface the surface we call the nanopillar surface. On the right surface, we have flat. And so this substrate itself is just quartz, or you can consider as a glass silicon dioxide. On the nanopillar area, what we do is we use electron beam lithography to make, to first create metal masks. And where there's a mask, when you do the etching, when there's a when we do reactive iron etching step, those places the material cannot be etched away, and all the rest of the places the material are etched away. The end result is it creates these vertical nanopillars. In this substrate, on this substrate, the nanopillar is made of silicon dioxide. The flat area is made of silicon dioxide, and on the right area there's no mask, so the whole area is uniformly etched down. So it's just flat silicon dioxide surface. So you can see that we have areas, each of the square here is probably by 100 by 100 nanopillars, and on the red side there is no nanopillars, but this is both the nanopillar area and the flat area on the same substrate. And when we put, for example, in this case, hippocampal neurons on top of it, some of the cells will randomly land on the nanopillar area, some of the cells on the flat area. By bright field imaging, you don't see a huge difference. But if you look at the fluorescence imaging, we are using immunostaining of the tau protein. Tau protein is quite specific to neurons. And 20 hours after we plate the hippocampal neurons, you can see that the cells on the left side on the nanopillar area already grow out very long axons and dendrite. They finish axon dendrite differentiation. And the cells on the right side, on the other hand, is mostly round, has some very short and immature neurites. So in biology, particularly neurobiology, this is a system very well studied on glass surface. Hippocampal neurons, embryonic hippocampal neurons, usually take 48 hours to 72 hours to differentiate. So they usually start with a number of short neurite, and they compete, and over 2 to 3 days, and one of the neurite win and become an axon. The rest of them become dendrites. And this image was taken only 20 hours after plating. So you can see that on the nanopillar areas, the neurons are growing much faster. They finish axon dendrite differentiation much faster compared with neurons on flat substrate. It's shocking, a contrast to us when we first see this result, the cell behavior result. And again, I want to stress that the difference, they are inside exactly the same culture, starting from the same batch of cells, all made of silicon dioxide and all coated with the same cell adhesion molecule. The real difference between the two different areas are just their topography. One is smooth and one has nanopillars. And now the question is why? Why cells on nanopillars actually grow out axon dendrite much faster? To understand that, today I want to talk about, actually I don't think I have time to talk about the third topic, but mainly about the first and second topic. I want to start with how we using ultrastructure analysis to look at the nanobio interface, exactly how cells interact with nanostructures by electron microscopy. So how cell membrane interact with the topography, protrusions, or indentations. For example, if you have surface with protrusions, and if you have a surface with indentations, when you grow cells on top of it, some people think the cell membrane is very plastic. That means it's easily deformed, and they could completely deform and adapt to wrap around the nanopillars and also growing into the nanoholes. And because the cell membrane not just lipid, they also have a layer of actin and cortex, so that gave it a certain strength and tension. So, and some modeling considers cell membrane as elastic material. If it's elastic, it could form, a tent on nanopillars or a tent on nanopores. So exactly how cell membrane interact with different topography turns out is not very well understood. And to look at the interface, because those nanostructures are often in the order of, tens of nanometers to hundreds of nanometers. And so we decided to use the electron microscopy to look at the interface. We started by using transmission electron microscopy. And in the transmission electron microscopy, we have to, we fix the cell, we infuse the cell with resin, and we polymerize the resin. And a lot of times those nanopillars or nanostructures are actually, you cannot cut into thin slices. So we have to using chemical method to etch away the glass using hydrogen fluoride. And then we slice them into backfill with resin and repolymerize and slice into thin slices and put onto the TM grade. I have to say this is a really pretty difficult process, and after a couple of days of immersed in the resin, the image quality is usually pretty bad. You can see the intracellular structure is not very clear, but you can still see the cell membrane wrapping around the nanopillars. So the cell membrane are not tenting on the nanopillars as the elastic membrane usually do. We tried the nanopillars of a different geometry. Some are very tall, some are very thin, and some are big. And usually, as long as the nanopillars are very sparsely distributed, we see the cell membrane wrapping around it. We could not, using the same method, to do holes, because after we etch away the substrate, we actually don't know where the holes are. And it was later, a postdoc, Francesca, came to the group. We decided to use a totally different technique to look at the nanobar interface. In this case, we use FIB-SEM. It's called the focused ion beam scanning electron microscopy. So to use FIB-SEM to look at the nanobar interface, the first thing we need to do is we need to embed the cell in resin, but in a very thin layer of resin, not like a TM resin, which is a big block and requires thin sectioning. In this particular case, we first fix the cell, and then you add the resin, and the resin is going to be immersed into the cell and also outside the cell. Then what we do is we do a quick spinning, and then resin wash to wash out the epoxy resin outside the cell, but keeps the resin inside the cell intact. And then we polymerize the resin. Here is an example of the 3 cells already embedded in the plastic resin. Of course, during this process, we also do heavy metal staining to give contrast to membrane and also to proteins. And you can see it really preserved the structure well, and most of the resin outside cell are removed. This is again an image of a cell on nanopillars. And after thin resin embedding, again, those are the cells embedded in resin. And you can see the fine features here. It's very well preserved. From here, you already can get a sense that, cell membrane extend out and grab onto those nanopillars. And when our group first started looking at the cell, how cells interact with those protrusions, we initially just hope cells don't die. They don't poke through the cell membrane and kill the cell. It turns out most of the time, the cell really likes those protrusions, and they extend their membrane and grab onto it. So to do FIB-SEM imaging and after we see the SEM image of the cell, and we see we want to look at the interface, particularly align this line of nanopillars. What we do is we come in with high energy gallium beam. The gallium beam is going to bombard this line, along this line of the surface. We will bombard and remove any material and to create a trench. And this trench will expose the interface between the bottom cell membrane and the nanopillars. And then after creating the trench, we can use SEM to look at the interface. And this is how it is done. And again, we start by identify this is a neuron on nanopillars, and those are the neurite. So we cut through the cell body, which is this surface here. We cut through neurite 1, cut through neurite 2, and cut through neurite 3. And this is the image for the cell body, and this is the image at the corner between the cell body and the neurite. And this is the neurite 3. The FIB-SEM gave amazing contrast and resolution. I have to say much better than what we had achieved using TM, transmission electron microscopy. For example, this is a SEM image of a cell on the nanopillars. On top of the cell is a very thick layer of platinum to dissipate the electron charge effect. SEM usually have the bright area on metal stand membranes. But if we just simply do black and white inversion of the image, it looks just like a TM. It's really nice. And then if you focus this particular nanopillar and zoom in on here, and this particular nanopillar, you can see the cell membrane, wiggles on the flattened membrane and wrap around the nanopillars. And this is the ER, wrap around nanopillar pretty tightly. And those are mitochondria and ribosomes in the cell. So again, what we see is that cell membrane usually wrap around pretty tightly along nanopillars, even more tightly so than a flat substrate. Again, you can see different shape, and it can even deform the nucleus, deform the cell membrane. And using this technology, we can easily look at how cells interact with holes. Here are the 5 nanometer diameter. This is 500 nanometer diameter, but 1 micrometer depth. When you look at the holes, you can see the cell membrane does not grow deep into the hole. Instead, it's trying a little bit to grow into the hole, but really not attaching to the surface of the hole. Even a shallow and a wide hole like those ones, like this one, we can see the cell membrane touch the bottom of the hole on some places, but again, not conformally deform the cell membrane to fully fill in the hole. And we measure the cleft distance between the cell membrane and the substrate. What we can see is for nanopillars, usually the distance is much smaller, on the order of 20 nanometer, 15 to 20 nanometer. On flat substrate is usually 50 to 100 nanometer. On the holes is very far because they're really pretty far away from the substrate. So that gave rise this very interesting observation. It's an asymmetric response. So for protrusions, the cell membrane wrap around very tightly, but for indentations or holes, the cell membrane does not grow into the hole. So from this study, you can see we cannot see the cell membrane as an as a very simple object. It cannot see it as a plastic or elastic. It responds to different topography in a very different way. Again, that gives rise to the question that we are really interested in: how cells sense surface topography. It has been like a 20 to 30 years of research. In the beginning, people are believing that roughening the surface actually is changing surface chemistry. You're changing the surface energy or the functional group on the surface. And then later, hypothesis is increase the surface area. And there's also, control the more newer hypothesis due to control the spacing between adhesion site. I will not get into each of those hypotheses, but I can only say that none of the hypotheses are able to explain all the observations that exist in the literature. Vast amount of studies on how cells are growing on different surfaces. So here I go to the second half of my talk is we really delve into the mechanism of how cells sensing different surface topography. From our our electron microscopy study, we know that for nanopillars, when the cell wrap around it, it locally deforms the cell membrane, pretty severely deforms the cell membrane. and because it had to wrap around very tightly around it. So if we hypothesize that when you deform the cell membrane, it will trigger certain signal pathways that flat membrane does not. So the first intracellular process we look at is endocytosis. And many of you probably heard of endocytosis, particularly Clathrin-dependent endocytosis. This is one of the key cellular processes, cells, sensing outside signal and even, growth factors and eating. This process is conserved all the way from yeast to human. So Clathrin-dependent endocytosis start usually from, a relatively flat membrane. And then as a protein assemble on the flat membrane, it deforms the cell membrane. And the curvature increases during the process until a vesicle form. And then Dynamin actually come and serve the neck and become a vesicle. So several key proteins involved in this process are actually curvature sensitive. And so we decided to look at the proteins involved in this Clathrin-dependent endocytosis. We started with Clathrin and Dynamin, two of the most widely studied proteins in the Clathrin-dependent endocytosis process. Here is an immunostaining image. Each of this black dot here is a glass nanopillar. Again, the nanopillar is made of silicon dioxide. It's vertical. When you take a 2D image, it appears as the dot here. And that's a Dynamin image, that's a Clathrin image, that's an actin image. Dynamin image, you can already see some of the green dot colocalized with the nanopillars. Clathrin, you see pretty clearly they colocalize, although they have a high background. And actin is a lot clear. It's very clear actin accumulate on nanopillars. Here, when we first see this result, the question, there is some doubt whether this is real or not. The reason is, and it's a three-dimensional, nanopillars are three-dimensional structures. When you take an image, two-dimensional image, there's a projection effect. And also cell fixation may cause some proteins preferentially accumulate. To make sure this is indeed a curvature effect, not some fixation artifact or the projection effect. What we did is we fabricate with nanofabrication to engineer a gradient nanopillar array. This gradient nanopillar array has, some big ones like 1 micrometer, 1000 nanometer diameter, smaller ones about less than 100 nanometer diameter. There's the same height, same spacing, and a range of the diameters can be covered in the same cell, by the same cell. Another, we use live cell imaging instead of fixed cell. In this case, the cells are transfected and expressing a Dynamin GAP construct. And then we took a live cell imaging, and then we average the image, live cell imaging. And what you can see is the result is quite striking. For large nanopillars, you can see very clearly here, there's not a lot of protein. There's still some protein accumulation, but not a lot on the nanopillars. But on the other hand, for the smaller diameter nanopillars on this range, if you look at the bright field image, you barely see, particularly the small ones, there's so small bright field image, you cannot see clearly, but you see very strong protein accumulation on those nanopillars. So this result quite conclusively show us it's a size. So whether proteins, and in particular, this is in the same cell. So whether protein accumulate or preferentially accumulate those nanopillars depend on the size of the nanopillar. And so we averaged the many cells. And then we also did, we did a 3 color. One is we image the plasma membrane, we image Clathrin, and we image the Dynamin. 32 different diameter of nanopillars. And then we normalize the protein distribution against the lipid distribution, which is a membrane surrounding the nanopillar. Because when you do the two-dimensional projection, there are more membrane on nanopillar locations than the flat locations. So after we normalize against the membrane intensity, what we can see is this kind of a distribution. So Clathrin and Dynamin, they start to preferentially accumulate on the nanopillar locations only when the diameter of the nanopillar is less than around 400, 500 nanometer. Above 400, 500 nanometer, cells view those nanostructures as flat. They don't see it. At least in terms of Clathrin-dependent endocytosis, they don't see it as a curved membrane. So the curvature range is around 450 nanometer. Using nanofabrication give a lot of flexibility. So using nanofabrication, we can also make those nanobars. Those nanobars, you know, if you think about it, it's like more like a book you're standing on a table. So at the end of the nanobars, it's high curvature. On the middle is relatively flat. We are imaging relatively around the middle of the height. And if you look at Dynamin GAP, it's such a strong contrast. The protein preferentially accumulate to the end of the nanobar with high curvature, much less along the middle, whereas low curvature. And of course, we look at, you know, many different proteins and membranes and see how they distribute around nanobars. Nanobars give a really good control because it's the same bar in the same cell, give different, two different curvatures. For example, for cell mask is a lipid dye staining the membrane. What we see is the whole bar light up. That means the membrane wrap around the nanobar pretty nicely. When we look at this protein called mCherry-CX, again, the whole bar light up. That means this membrane protein uniform distribute along the nanobar. But when we look at the protein that's more specific to Clathrin-dependent endocytosis, that's including F-CHO, transferrin receptor, epsin, amplifying, Clathrin, and Dynamin, and AP2. You see, a lot of times those proteins are like dumbbells. They only accumulate to the end of the nanobars. Very strong bias towards the end of the nanobars. That's, you know, this study shows us that it's a curvature-dependent process, and those proteins involved in Clathrin-dependent endocytosis preferentially accumulate to highly curved locations. And those highly curved membrane locations are not static. So those proteins will come and go. It's a typical, you know, Clathrin first accumulation and then Dynamin come in to finish an endocytosis process. This is a typical Clathrin-dependent endocytosis. So they are not just recruiting proteins, they are actually activating the entire process. Okay, so that's the first cellular process. We look at the Clathrin-dependent endocytosis, and I already showed an image earlier. During the study of the Clathrin-dependent endocytosis, what we noticed quite interesting protein is actin. So actin, most of the time, if you see the actin staining, they're just fibers, they're cytoskeletons. But when we put the cells on nanopillars, we see strong accumulation of actins on nanopillars for a reason that we don't know. And if you look at the nanopillar of different diameter, 180, 400, 700, you see more clear protein accumulation on small diameter nanopillars than large diameter nanopillars. So we decide to look at whether this actin accumulation is dependent on the membrane curvature. So if you look at the nanobars, again, this is a cell growing on the nanobars. If you do the actin staining, you see the actin staining is still show those stress fibers, fiber-like structure, but on nanobars, again, the actin is on the two ends of the nanobars, not wrapping around the entire nanobar structure. And when we look at the CUI structure, you see the I, then the actin is on the two end. But if you look at U, for example, it's interesting. U is actin is on the outside the U, but very little in the inside of the U. And we will later show they really strongly prefer positive curvature because the membrane inside the U is actually a negative curvature. So when we see the actin on nanopillars, the first thing hypothesis we have is maybe those actin facilitate, cell adhesion. Therefore, we decide to look at adhesion molecules instead of myosin, paxillin, and vinculin. In each of the pair of the image are the same location, same cell. And this is the actin and this is the myosin. You can see that for stress fibers, they colocalize very well. Wherever there is an actin fiber, you see the myosin. But on the nanopillars, you see very clear accumulation of actin, but there's no myosin 2. And the same thing here. When you see accumulation of the F-actin here, you don't see paxillin, and we don't see vinculin either. So those actins are not involved in, at least not involved in mature focal adhesion. So we decide to look more into the type of actin in the cells. And generally speaking, there are 2 types of actin fibers in the cell. One type of actin fiber are those linear, very long linear actin fibers, mostly stress fibers. And there are another type of actin, so-called branched actin. So this is a linear actin, and those are branched actin. Most of the branched actin on the cell edge. And linear actin and branched actin, if you stain actin, they both show up. But these 2 types of actins are actually polymerized by different protein initiators. The branched actin are polymerized by up-to-3 complex, and the linear actin are polymerized by formin. And formin produce linear actin. So we decide to look at how does the actin initiator distribute along the nanopillars or nanobars. So we transfect cells with UP3 GFP here, and those are the nanobar arrays, and the 3 cells here. You can see UP3 GFP are mostly cytosolic and appear as a smeared background, but on nanopillars, they show very strong signal towards the end of the nanobar. Sorry, on nanobars, they show strong signal at the end of nanobars. In all 3 cells, they show this behavior, very little around, along the middle. And so when we look at the formin, so this is a formin. Formin again polymerized linear actin. What you see there is almost no formin on the nanostructures, on the nanobars, either to the end or to the middle. So this one really, allow us to realize the actin that on our nanopillars or end of nanobars are actually initiate or polymerized by up-to-3, and so therefore they are branched actin. So we start from actin, and we find out up-to-3 are its initiator. So up-3 are not curvature sensing protein. Actin itself is not curvature sensing protein. Up-3 have 2 activators, N-wasp and cortectin. So when we look at the N-wasp and cortectin, both of those one show preferential accumulation to the end of the nanobars. And both the N-wasp and cortectin can be activated by FBP17. So going from down, up, down is actually going upstream. And FBP17, we look at FBP17, show clear accumulation to the end of the nanobars. And by the time we find FBP17, we know that we probably hit the target, because FBP17 is actually a well-known curvature sensing protein. It has a family of the 3 proteins. We examined the entire family. All 3 proteins show preferential accumulation to the end of nanobars. So FBP17, it's so curvature sensitive that it's extremely clear. In this particular experiment, we made this wedge nanobar. On one end has sharp curvature, and another end has lower curvature. When you look at the FBP17, you see those wedged bar has this is a small end, this is the big end, and there's much more protein on the small end than the big end. And similarly, we made a gradient nanobar. So this gradient nanobar, they are the same length and height and the spacing. The only difference is their width. Their width determines the curvature at the end. So you can see this is a very sharp nanobar or narrow nanobar, and those are wider, 100. This is 1 micrometer wide. This is 100 nanometer wide. And we also use immunostaining of the endogenous FBP17. You can see that on small diameters, you see the preferential accumulation of the proteins, but large diameter, you see very little accumulation. And when we plot out the FBP17 and the actin, they show approximately similar cutoff, and again, also similar to the endocytosis, around 400 to 500 nanometers. And beyond that, and there is no preferential accumulation. Only when the nanopillars are smaller, they show preferential accumulation. Again, you know, to validate that the FBP17 is really the reason why actin preferentially accumulate on the nanopillars or high curvature locations. What we did is we construct a dominant active FBP17. And so FBP17 has 3 domains. F bar domain is a sensing membrane curvature. It's a very well-known curvature sensing protein. And HS3 domain actually HS3 domain are the sensing domain where you know induces the activation of downstream signaling. So we chop off this SH3 domain and keeps the curvature sensing domain. And in this particular cells, a dominant active FBP17, this is a wild type FBP17. So wild type FBP17 accumulate on the end of the nanobars, and also protein actin accumulate end of nanobars. This dominant active FBP17, on the other hand, also accumulate on the end of nanobars, if you see here more clearly, but there is very little actin accumulation on the nanobar end of nanobars. And this experiment really confirms that why actin accumulate on the high curvature locations are due to this family, FBP family of FBP17 family of curvature sensing proteins. So in the last 10 minutes, I would like to the last 10 minutes, I would like to revisit the initial observation here. And so when we study the endocytosis and the actin accumulation, those are more of at the you know, cellular level or protein level. Ultimately, we want to understand the cell behavior. For example, why neurons grow out longer neurite and grow faster on nanopillars compared with flat area. Again, we could not tackle this problem. The observation was that we see this phenomenon at least 5 or 6 years ago. We don't understand it until very recently. So what we believe is that this nanopillars or nanotopography, it alters how cells see the substrate, and particularly substrate stiffness. So our nanopillars are very rigid structures, and the substrate itself is glass. Nanopillar is glass, is extremely rigid. As I mentioned that, you know, nanopillars induce actin accumulation around the nanopillars. But if you look at a zoomed out image on a large area, and this is actin distribution on the flat, and this is actin distribution on the nanopillars. When the nanopillars are very dense, that means there are a lot of nanopillars underneath the cell, not just a few nanopillars. We see a dramatic reorganization of the actin cytoskeleton. So on the flat areas, you see most of the actin as those strong stress fibers. And on the nanopillar areas, they accumulate on the nanopillars, but those stress fibers are largely disappeared. So there's a drastic reorganization of the actin cytoskeleton. So how this reorganization of cytoskeleton affect the cell behavior? And so we look at cell area and YAP, which is a mechanosensor. We use it as a mechanosensor. So we decide to compare 3 surfaces. One is a flat quartz substrate, one is nanopillar quartz substrate, and one is hydrogel. The reason why we we decide to look into hydrogel is because here, when we see that most of the stress fiber are largely disappeared, we know that when cells are cultured on very soft substrate, they do not have a lot of stress fibers. So we decide to compare with the nanopillar quartz versus the hydrogel substrate. And you can see that on the flat substrate, and first of all, the cells are very much larger, actually significantly larger than the cells on nanopillars or the cells on hydrogels. And if you look at the YAP staining, on flat substrate, YAP are mostly on the nucleus, while on the nanopillars or on the hydrogels, YAP is more or less, uniformly distributed, but not only in the nucleus, but also in the cytosol, which is a result of the cell, mechanobiology. The cell actually seems much, softer than the cell on the flat substrate. This is a zoom out. You can see the cell YAP is a more uniform distributed, while on the flat YAP is more on the nucleus. And we actually using AFM, the atomic force microscopy, to directly measure the cell stiffness, the apparent cell stiffness. And so this is a apparent Young's module of the cell. That's a flat, and this is nanopillar, this is hydrogel. So we can see that both nanopillar and hydrogel are substantially lower than the cell on flat. And when we try to pull a lipid tether from the cell membrane, there's a tether force. Again, both nanopillars and hydrogel are lower than the flat substrate. So for this particular study, I want to emphasize the nanopillars are made of glass and quartz, and so the stiffness of the nanopillar substrate is on the order of probably 100 gigapascal, and hydrogel in this case we're using is 1 kilopascal, and so those are like almost 6 order magnitude difference in substrate stiffness, but they induce similar cellular responses. And then we look at the neurons, which we talked about before. So neurons on the substrate after 20 hours are mostly, round neurite, but neurons on nanopillar substrate, a lot of them grow out long neurite. And interestingly, neurons on hydrogel surface also grow out long neurite. And actually this result, when we started the experiment, a paper hasn't published, but this result is independently validated by a very recently published eLife paper. So it's very interesting that on flat substrate neurons, very few neurons grow out long neurite, but on nanopillar and hydrogels, really a lot of neurons grow out long neurite. So all of those are the neurons. Then we decide to look at another cellular behavior, which is stem cell differentiation. So HMC stem cells differentiate into bone cells. We can use different protocols to mix them into differentiate into bone cells, or osteogenic differentiation of fat cells, dopogenic genic differentiation. And you can, I'll skip the description, but you can see that on hydrogel on nanopillars, there's cells are much less likely to differentiate bone cells than flat, and which is opposite for bone cell for fat cells. Fat cells are soft cells. Bone cells are more rigid cells. They prefer more rigid substrate. So that's a final of my talk. Is from all of those studies, and it gave rise to, we proposed the curvature hypothesis, and that says the membrane curvature at the cell membrane interface can directly serve as the biochemical signal that translates surface topography into intracellular signaling. So we believe membrane curvature is either the mechanism or one of the very essential mechanism of a whole cell sensing surface topography. And with that, I would like to thank the people who did the work. My group at Stanford is really a very talented group, and the work I covered today, some of the people already left and started their own research group. Francesca is now a professor in Italian Institute of Technology. Wenting is now a professor in Nanyang Tech at Singapore. And Zinab is now a professor at UC San Diego. Thank you. Questions?
Great. Thank you so much, Dr. Cui. That was a fascinating and beautiful talk too. All of those images are so extraordinarily crisp and clear and really awesome. The floor is open for questions. If you have a question, please type it into the chat so that I can read it to our speaker. I'll start first. You showed that you had the differentiation of adipogenic versus osteogenic cells, but you also showed that you had some integrin independence to this curvature phenomenon, and that integrins, like mature focal adhesions, weren't required for your phenotype to be seen.
Mm-hmm.
So do you think that that means that some of this differentiation and perhaps maturation of the neurons is not driven necessarily by integrin signaling?
I would not get that conclusion. So what we see is the actin accumulation on the nanopillars do not have, the myosin 2 or the pacillin, those proteins. But what we see is that, the fact that when there are a lot of actin accumulating on nanopillars, it reduces stress fiber. It actually reduces focal adhesions. It significantly reduces focal adhesions. So it does affect integrin, but in a negative way.
Okay, great. Thank you. GQ?
Yeah, that's fascinating. And yeah, I learned so much today. So just a very general question that, have you, is it possible to analyze the lipid molecules or cluster or spingle myelins and these lipid molecules in the curvature? Is possible or to do that?
Yes, it is possible. We actually have some preliminary result on that, but it's not. Lipid diffuse very fast. The preferential of lipid there are many due to proteins that make those lipid.
So how this increase the branched actin versus the linear actin, and because sure. Clearly very different patterns, and how those global changes of cytoskeleton affect the cellular behavior differentiation process. So because differentiation means that, and from certain gene expressed, then so when they differentiate, they are turned a differentiate cell feature, which that, how globally overall this cytoskeleton structure induce different signaling into the nuclear program, which make this biological differentiation kind of. So
that's I can speculate. That's a really good question. Here, this is I guess the image you talked about is a really striking reorganization of the actin cytoskeleton. So I think actin cytoskeleton is clearly involved in differentiation, and in our comparison study of nanopillars in hydrogels, in particular, for example, if you grow cells on hydrogels, they do not have this flat substrate either. They do not have a lot of stress fiber either. So how the differential, how the stress fiber disappearance involved in the stem cell differentiation, that I would say it's probably involved in many proteins, and clearly, as MK indicated, it does involve integrin signaling, and I would say a lot of mechanobiology signal pathways are changed in order to ultimately contribute to the stem cell differentiation. I would not say one or the other, but most likely many are changed.
So when that's great. When the normal tissue are injured, they form fibrosis, fibroblasts, fibrosis, right? So
Yeah.
the tissue become more rigid, right?
Yeah.
And then we see a lot of, like nanoparticles, injection sometimes have some benefit effect, induce tissue regeneration, normalize tissues, right? Seems to show some benefit. We don't know what it is. Some, now even exosomes, for example, circulated is kind of like the size is like in the nano scales, right? Like 100 nanometers, exactly like your nanopillar. So my question is, what do you think in general, if I just inject any nanoparticle, any small nanoparticles, any composition, and inject into the injured tissue where the fibrotic tissue is developed, and would that be beneficial or explain some benefit effect we observed?
I have a speculation for this particular problem. Actually, we have a hypothesis. In this particular case, for example, why nanopillars significantly reduce the stress fibers? One of the mechanism is actually endocytosis. So because membrane curvature induces endocytosis, and this endocytosis actually disassemble focal adhesions. And so for your question, even you're using nanoparticles instead of nanostructures, but people know that a good size depends on the size of nanoparticle. But if it's like around 50 nanometer or 20 to 50 nanometer, cells love those nanoparticles. Too large, they don't particularly like it. But it's around maybe 20 to 100 nanometer, cells just love those nanoparticles, no matter what they are made of. So that triggers endocytosis. And I don't take responsibility for this hypothesis. My hypothesis to your question is most likely the triggered endocytosis, enhance endocytosis by the cells, and will make the cells soft and
so
potentially help the.
So evolutionarily, the neuron are not necessarily endocytotic cells, right? Macrophages are, dendritic cells are, right? So why would clearly the evidence is there. So evolutionarily, why is that?
Why neurons are not endocytotic cells?
Like a cardiac cells, right? They're post-mitotic, they are very pretty much functionally specialized, right? And they need this kind of signaling.
Yeah, neurons are quite special. They do endocytosis at synapse. They need really fast endocytosis.
They do, okay.
Yeah, and but they usually, they don't divide. So they probably endocytose and less than other cells. I actually don't know that.
Yeah, we underappreciate that, the phagocytotic activity. Yeah, absolutely. Thank you. Great.
Thanks, GQ. Do we have any other questions from any of our students today? It does not look that way, but I want to say thank you again so much for taking time out of your schedule to log in and share all of this work with us. Like I said, just fascinating and really cool work. We've got some claps and some thumbs up emojis going through the chat. So thank you so much. I actually have to log off now because I have to go take care of something in the lab that just came up, but I will leave you in Carrie and GQ's hands, and I'll reach out to you to follow up in a couple of days. So thank you.
Thank you. Okay. Yeah. Again, thank you very much for asking question, for the great talk, and that also I have another meeting I have to go. And
okay, thank you.
Yeah.
Thank you all.
Thank you.